The WHO estimates that about 1 in 6 couples are struggling to conceive, and this of course can be very distressing, as conceiving a child can be a very big part of their life. So it's a very important problem, and this can come from environmental effects, but also genetic effects, which are sometimes not very well understood. Chromosomes store genetic information. In humans, generally, we have 23 pairs of chromosomes, and sex chromosomes are different between male and females.
So back in the 80s, a group of scientists discovered that when they looked at the chromosomes of men that were infertile, they often found that one of the chromosomes in the genome of those individual men contained a missing piece for one of the particular chromosomes, the Y chromosome. And this was the first evidence that the Y chromosome might have a particularly important role in the genetic causes of male infertility.
So in this study, what we really wanted to discover was the function of individual Y genes. So we knew that the Y chromosome was important for fertility, but we didn't know exactly which genes were playing a role and how. To do this, what we did is using genome editing techniques like CRISPR-Cas9. We removed genes one by one from the Y chromosome. We then made mouse models that were missing certain genes. Once we had these mouse models, we studied all aspects of their fertility to really understand how the absence of this gene or the absence of this combination of gene can have an impact on fertility. Some of them were fully fertile, so we didn't see any changes in their fertility. Some other were subfertile, which meant they were still able to produce offspring, but they were less efficient at doing so. There were some other issues during reproduction. Other mice were completely infertile. And then there were also mouse models where we saw that we only saw infertility or subfertility when we delete multiple genes at once. Deleting one gene might not be enough to see an effect in the animals, because another gene is compensating for the absence of that gene which we just mutated. So starting to remove more than one gene at a time is very helpful, because then we can really understand what this type of gene does.
It's good to mention, first of all, that knowing what different genes on the Y chromosome do is very important, and for men that have deletions of the Y chromosome, knowing why they're infertile is very important to them. In terms of therapy, we're a long way off developing any technologies for reproduction in men with deletions of the Y chromosome. You could conceive of a method whereby you could generate sperm from stem cells in a dish. That's currently not possible actually for any mammal, including humans. But if that were possible in the long term, one could conceive replacing those missing Y chromosome genes into that Y chromosome in a dish, and ultimately in doing so generating sperm and potentially offspring from those sperm. For many, many years, people have focused on the function of the Y chromosome in the germline and in male fertility. But I would say the next generation of sex chromosome biology research is to ask what the Y chromosome is doing actually outside of the germline, so other functions in the body. Now, why is this interesting? Well, we know from recent research that as men age, their cells gradually lose their Y chromosomes. We don't know how this happens, but we do know that men that lose their Y chromosomes have an increased chance of developing other diseases not related to infertility. So examples include cancer, diabetes, and Alzheimer's disease. So what this is telling us is that the Y chromosome is holding other secrets. It's doing important things in other tissues of the body. And so what we would like to do, now that we have this selection of mice that contain deletions of different genes on the Y chromosome, is to now look outside of the germline at other organs, for example, the brain and the heart, and to ask, do these mice exhibit abnormalities in other tissues? And if they do, we could begin to understand what these Y genes are doing in other tissues to maintain adult health.
What I really enjoyed was working in a field that very few people are working in. So any discovery, even if it's a small one, is something completely new that nobody has ever seen before. So that's very exciting.